Okay, this is your pre-lab video on the hydrolysis of gelatin lab. In it, you're going to be comparing 4 different enzymes, bromelain, papain, actinidin, and then ficane. And you're going to be doing so by quantitative means. Now, this is important. You need to have some numbers to go along with your um, lab, okay? Because you're going to be making a hypothesis about which of these 4 do you think would best hydrolyze the gelatin. Now, in doing so, since you have to, I don't want you to just randomly pick one. I want you to have some background. So, what you should be doing as part of your pre-lab is you should be looking at these 4 enzymes and determining, and also the structure of gelatin, and looking at them and saying, okay, well, which one of these 4 enzymes from 4 different fruits is going to determine or what's going to cause the best hydrolysis of the gelatin. Okay, now the materials that you have available to you, you have test tubes available to you, you have beakers, petri dishes, a ruler, or many rulers if you want, balances, and then distilled water, and you're also going to have a gelatin solution, which is going to be about 3 to 4% mass over volume. So those are the materials you have. If you want additional materials, if you think of something else when you and your group members are deciding your plan of action, which is the key part of this, you guys need to know what you're doing before you actually do it. Then if you know what you need, by all means ask me and I'll see if I can find it for you. Okay? But you're going to have 4 different fruits. Okay? Your research here, okay, from these 4 different enzymes should tell you where these fruits or where these types of fruit can be found. But considering that, for example, bromelain is found in many different fruits, I'll go ahead and let you know what your 4 fruits are. You just have to kind of match them up as your main source of that enzyme. So your 4 fruits, okay, you're going to have figs, you're going to have pineapple, you're going to have papaya, and you're going to have kiwi, or kiwi fruit. All right, now I'm going to make concentrated solutions of these 4 guys, and hopefully in your research you should be able to associate one of these enzymes with one of these particular fruits. So that's one little bit of homework you're going to have to do. But again, the main issue here is that you have a game plan with your group members, okay, and that you know something about these enzymes and these fruit and these different types of fruit, specifically the enzymes, and a little bit about gelatin and how gelatin forms and how those peptide bonds form, right, and how gelatin sets. Because all 4 of these are going to affect how gelatin sets. What you're going to do is, again, by quantitative means, determine which one does it best, which one's going to prevent the setting of the gelatin the best. All right? So, again, I cannot emphasize this enough because this lab is easy. Easy, 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 easy. But this is a little more interesting if you have to do a quantitative lab here. Okay, it's not by any means beyond you. It's just usually if this is usually a qualitative lab, but in this case I want numbers. So you can, however you in your group want to figure out how to get those numbers, then more power to you. I would like to see your procedure and your idea first, as well as your hypothesis. Again, your hypothesis needs to be if and then. All right? So we'll talk about this a little bit more tomorrow. You're going to have 3 days for this lab. So like I said, any questions you have, make sure you get them out tomorrow.